Good evening and welcome to the EKU Alumni Spotlight Series. I'm Dan McBride, Associate Vice President for Development and Alumni Engagement at EKU, and I am your host for our Alumni Spotlight Series. And tonight is a very special edition. We are teaming up with the Scholars Week Committee here at EKU and presenting a very special Scholars Week edition of the Alumni Spotlight Series. So if you are someone who is tuning in tonight because it's Scholars Week, we welcome you to the show and want to let you know that this is actually our 13th Alumni Spotlight of the year. We've been talking to different EKU alums since back in October, and we have a few more shows scheduled for later in the year. A couple of weeks from tonight, as a matter of fact, coming up on the 20th of this month, Dr. Deborah Alexander, who worked with the State Department for years and has an incredibly interesting career and story, and also actually did an internship with Mother Teresa when she was a student here at EKU. She will be our guest 2 weeks from tonight on the Alumni Spotlight Series. That show will also be at 7 p.m. We hope you can tune in for that. I'll tell you more about tonight's guest in just a few minutes, but we do have some other virtual programming coming up from the Alumni Office that I want to let you know about, things that are going to be coming up in the month of April, a very busy month of April for our virtual programming, and we'll go into May a little bit, and then we will kind of shut things down for the summer, take a little break, and then come back in August with some new virtual programming, so be watching for that. But here's what's coming up this week. It is At the Races. We can't do our normal Keeneland event this year because of limited capacity at Keeneland, so we're going to do some virtual online horse racing. That'll be Thursday night at 8 o'clock. EKU's At the Races. It's a fun game. It's a team building, our team work experience where you play on teams and bet on horses with fake money, and builds a lot of camaraderie. It's a lot of fun. If you haven't signed up yet, go and check it out on the EKU Alumni webpage. Also coming up in the next couple of weeks, we want to let you know about Giving Day. That is on April 14th, Wednesday, April 14th. We have a goal of 1,000 donors for that day. One day, one goal on the 14th of April, and we hope that you will join us and also make a gift to EKU that day to help us reach our goal of 1,000 donors. The next night, we have Alumni Bingo. It's our final round of Alumni Bingo for the year. We've done it a couple times during the year, very popular. A lot of folks have enjoyed it. That will be at 8 o'clock coming up on the 15th. And then our Colonel's Kitchen program, which is a joint venture with the Department of Human Sciences here on Applied Human Sciences here on campus. Dr. Dana Bush and myself host it with Chef Sang live from the Burrier Cafe, Cafe Burrier on campus. And we have a special treat coming up for the Kentucky Derby on the show this month when we'll be talking about some Derby favorites, and that show will be on April 28th at 8 p.m. So a lot of things coming up in the next few weeks during the month of April, a lot of virtual programming. We hope that you can join us for some of those programs, if not all of them. But as I mentioned tonight, we are teaming up with the Scholars Week Committee for a very special edition of the Alumni Spotlight Series. And our guest tonight is actually someone who is very qualified to be part of the Scholars Week discussion and to be the spotlight for the Scholars Week edition. Someone who was actually on our radar before and someone that we wanted to have as a guest on the show. And then when the Scholars Week people came to us and said, do you have an alum that you think would be great to be our alumni feature for Scholars Week? We said this is the perfect fit. Our guest tonight currently serves as an assistant professor in biomedical science at the Kaiser Permanente Bernard J. Tyson School of Medicine, holds a visiting associate position at the California Institute for Technology in the Department of Biology and Bioengineering. He got his Bachelor of Science at EKU in 2009 in biology. We are very pleased tonight to welcome to the Alumni Spotlight, Dr. Derrick Morton. Good evening, Derrick.
Good evening. Nice to be with you, Dan. It's great to be back at EKU, even if it's just virtual.
Yeah, so now you're in California, and we were talking earlier, this is one of those rare days where we actually have warmer weather here than what you have in California.
Yes, it's about 70 degrees here, actually. So it's very, very nice to see that it's 79 degrees, you said, in Richmond, in Kentucky?
Yeah, we had a beautiful day today. We've been fortunate this week. Of course, like most things in Kentucky, I'm sure the cold weather will be coming in the next few days. But we're enjoying the sunshine while we've got it.
Gotcha.
Now,
great.
And of course, you, of all people, know how Kentucky weather is. You grew up in Lexington, right?
Yes, I grew up in Lexington, born and raised.
So tell us a little bit about how you ended up at EKU, because I know initially EKU was not your first college choice, right?
Yeah, so at first I did enroll. I went to the University of Kentucky, and I really was, after that first year, I was searching for a bit of a smaller classroom and kind of, more interaction with peers and in those small classrooms. And so I came and did a summer course at EKU, and then I never left. And so I ended up finishing my degree at EKU and didn't go back to UK afterwards.
I know the story with a lot of the alums that I talked to. There were some influences in your life when you got to EKU, particularly one professor in particular that really made a difference.
Yeah, so as a really young biology student, I really didn't know a lot about scientific research, and a lot of students all want to be medical doctors, and that's kind of the thing. And so one professor, Dr. Michael Foster, who was, I believe at the time, the chair of biology, really kind of opened my eyes to the research field and science and all of the possibilities and the career that I could have. And so, yeah, he's really my first influence in actually research and getting into a lab and thinking about discovery.
Tell us a little bit about what you remember from your days at EKU and how things were on campus back then.
Oh, they were great. My first memory is I took a chemistry class in the summer, and it was a very small class. I got to know many of the students in the class, and some of them were still friends. And this is back in like 2005, 2006. And so one of my best memories was actually getting to know the students in the class, which was unlike my prior experience. And actually professors being very, very easy to work with, have conversations with, and that was largely due to the small class size.
As you think back, you mentioned Dr. Foster, of course, but are there other faculty members that you remember and other folks at EKU that left an impact on your life?
Yeah, so Suzanne Bird, Marsha Pierce are a couple of them that really were impactful. I actually did a small research project with Marsha Pierce while I was there that really kind of influenced me to go to graduate school eventually and do my PhD. And so, yes, Dr. Bird and Dr. Pierce are two faculty members I think about all the time.
Now, did you even work at EKU for a while toward the end of your time?
Yes, so I wanted to take some time off before going to graduate school. And so I actually worked in the dean's office, and I've seen a couple of them on here already. And so in the dean's office, I actually worked for Dr. Foster as well as Dr. Deborah Core as well. And yes, she's saying hello. Hello, Dr. Core, nice to see you. And then Dr. John Wade was the dean of the college at the time.
And from EKU, you went on to Clark Atlanta University. And I know that's another place where there was a particular person who had a great impact on your life and served as a mentor for you.
Yeah, so when I went to graduate school, my PhD advisor, Jadip Chaudhry, really had a really huge influence on my career. I was very successful in my PhD. I published well. And got several opportunities there. And so I really enjoyed Clark Atlanta University. It's an historically Black college and university in Atlanta. And so I really wanted that experience after leaving EKU. And it was a great time on that campus as well. And I had several mentors there as well.
Yeah, and I think that's one of the places when you really first started thinking about how you wanted to serve as a mentor for others as your professional career went on. Talk a little bit about how that came about.
Yeah, so during my PhD, I got the chance to mentor students in the lab. So undergraduate students that would come in the lab, I would train them. Then I got to teach a bit during my PhD as well. And so after that, I really wanted to continue that mentoring in research and teaching as well. And so I did my postdoctoral fellowship. And what's a postdoc? So quickly, a postdoc is a period of training after your PhD that prepares you for a faculty position. And so I did my fellowship at Emory and through an ERACTA program, which allows for mentoring and teaching along with your research.
Yeah, and another interesting thing about your time at Emory, and this is fascinating to me, this is when you had a chance to work with some monks and go to India, right?
Yeah, so part of my postdoc, Emory has this connection with the Dalai Lama, and so it's the Emory Tibetan Science Initiative. And so for about 10 years, every summer, a group of faculty and some postdocs from a couple of universities, so Caltech, where I have a visiting appointment, Emory and some other schools around the country, faculty would fly to a rural part of India where monks are actually in refuge. And so we would teach them science. And so they had a great interest in understanding science, and they have no formal scientific education. And so there were different groups, but I came in with the cell biology group, or kind of just the overall biology, and I taught genetics to monks for about 2 weeks.
That had to be an incredible experience.
Oh yes, it was transformative to really talk to the monks who had no formal science education, as I mentioned, and really just had no idea of even concepts of the sperm and the egg. And so we had very, really good, very rich discussion with them and understanding science. And I always say the monks were kind of some of the best students because their training, spiritual training, is very much kind of, I think, lawyerly. And so they debate a lot. And so they debated everything with us. When we presented an idea, they wanted the scientific evidence for it. And so it was a really, really nice way to teach science over the summer.
Now, I also know that during your postdoctoral experience at Emory, you won 2 service awards. Talk a little bit about some of the research and work that you did during that.
Yes, so I actually wrote several grants while I was in my postdoc. And so I have an NIH grant that I was awarded, which is a competitive grant. And then also I have a Burroughs Wellcome postdoctoral enrichment grant as well, which is a career development award, which is also a competitive grant. And so in my postdoc, I studied molecular genetics using the Drosophila model. And so I was interested in this neurodegenerative disease, much like Alzheimer's or Parkinson's disease. I was actually modeling those genes that are implicated in those diseases in the Drosophila fruit fly model. And so a bit later, I'm going to talk a bit about my research, but in my postdoc and now my faculty position, I use the fly model to study neurological diseases and genes that are implicated in those diseases.
Yeah, let's talk a little bit about how you get from Atlanta to California. How did that happen?
Well, actually, one day during my postdoc, I get an email from the Kaiser Permanente Bernard J. Tyson School of Medicine. And I take the call and interview, and it seemed to be a good fit at the time. And so I thought about it. And they're a brand new medical school, and they don't quite have the research outfit for my program. But I actually interviewed at Caltech, which is a premier research institution, and I was able to get a joint appointment for my research. And so after that, then I signed on.
I know also part of what helped you make your decision was their emphasis on some of the diversity issues and initiatives that they are interested in. Talk a little bit about how that interests you.
Yeah, so all throughout my career thus far, I've really been big in diversity, equity, inclusion, and STEM. And so I've actually just was funded through the Burroughs Wellcome Fund to start a pipeline pathway program for underrepresented students in science and medicine, which will launch this summer. And so that's been very, very important to me, and central to Kaiser Permanente School of Medicine's mission is this diversity, equity, inclusion, and using DEI in the mission to really level the playing field in who is actually going to medical school, and then also how we can impact healthcare in a larger way with some of the disparities that we see in medicine.
Yeah, and I know also the teaching part was something that was really important to you. You wanted to be able to continue teaching.
Yes. So I got the best of both worlds. So I get to teach, and I also get to do my research as well. So I'm primarily teaching at the School of Medicine, and my research is at Caltech.
Now, you've had a couple of pretty impressive recognitions. I know MIT in 2018 called you a rising star in biomedical. You were on the list of 100 inspiring Black scientists in America in 2020 as a rising star. So, that kind of has to feel good to get that kind of affirmation and support for the work you're doing.
Yeah, definitely. I always think about that, and I'm very happy and very proud of those accomplishments. And I'll say this: I like to always say that I'm just one example of what I like to call Black excellence, and I think it's important that there are many more of me out there, and they are just needing the opportunity to showcase their strengths as well. And so I am very proud of those achievements, but there are many, many more of me, and just looking for an opportunity. And so in my career, I hope that I can play a role in providing those opportunities as well.
Well, for those folks who have tuned into our Spotlight series episodes before, we're going to do something a little bit different tonight because we are partnering with Scholars Week, and we're going to let Dr. Morton do a little bit of a presentation on his current research. And I'm going to be honest, you and I had talked about this last week when we were chatting, and I don't understand half of what you're saying, but I understand that the work you're doing is important, and you can talk a little bit about why it's important and kind of explain. So I'll kind of turn it over to you and let you talk a little bit about the current research you're doing.
Good. And I hope to make it digestible for everyone on the call as well.
All right. So we'll ask our guys. There we go. Okay, we're ready.
Good. So as I mentioned earlier, I am actually using a Drosophila fruit fly model to model neurological disease. And so first, why am I using this model organism? I think it's important to talk about that first. And so in science, when we want to understand how things are going awry and causing dysfunction and that lead to disease, if you have the neurological disease, I can't ask you to donate your brain, right? There's not many people that are going to say, well, you can use my brain to study it. And so what we do, we use evolutionarily conserved organisms to study particular genes or processes that can give us insight into what's actually going on in human disease. And so that's exactly what I'm doing with my model of Drosophila, looking at neurological disease. And the Drosophila model is really good for looking at genetic interest. And so the disease I'm going to talk about today is an autosomal recessive disease that is a genetic disease. And so the Drosophila model is really nice for that. And on the other side, I am also going to talk about this complex called the RNA exosome. And so I am interested in how genetic material, when it has defects in it, can cause particular pathology in neurons. And so I'll go through that a bit as we move forward in the talk. Good. And so before I move forward, I really wanted to highlight, since mentoring and teaching is a large part of my research career, I recently just published an article in PLOS Genetics that had a host of high school students, undergraduate students, and graduate students. I really wanted to highlight them because they are in part responsible for us getting this publication out. And they were actually, a lot of them, for the high school students and especially the undergrads, this was their first publication. And so we had a great time doing this project, and I just wanted to highlight them here. Okay, and so now it'll get a little bit in the weeds for those who may not be scientists, but I hope to keep you with me. So I like to start out with this kind of key foundational question. So how does one set of genes give rise to a very diverse array of cellular functions? And so one thing I like to point out is this genome sequence, that every cell in our body has the same DNA blueprint. But how is it that we get this very diverse array of cells, right? And so the central dogma of biology is that we think about DNA flowing into RNA via transcription. And this is a process by which DNA is a very, very important molecule, or your genes are very, very important, that they have to have a process by which it has to be actually used as an intermediary to be read and processed And so your nucleus, in your nucleus, your DNA stays housed there, and the RNA actually is going to be transcribed and then go out into the cytoplasm, the other compartment of the cell, to where it's going to be used to be translated into protein. And so we call this the central dogma, or the genetic flow of information, DNA, RNA to protein. And our DNA is very, very important. And so it is housed in the nucleus and stays there. And this RNA kind of acts as a messenger to be used as a template for our protein synthesis. And so here, still, how do we get these diverse array of cells? We have the same DNA blueprint, we have central dogma, this genetic flow of information, but how do we get this diverse array of cells? And it really is the differential regulation of gene expression. And so that's a bit what I'm going to talk about today. So regulating these cells differentially gives this very diverse array of cells. And so while much of the regulation of gene expression is achieved at the gene-specific level, there are numerous post-transcriptional regulatory processes that occur. And so what is post-transcriptional regulation? So before an RNA molecule can be deemed competent to go out in the cytoplasm, the other compartment in the cell, and be used to make proteins, there are a series of modifications that actually happen to this RNA molecule. It's capped, it's spliced, it's further processed to aid in stability of the actual molecule such that it can be used for translation to make proteins. And so proteins are the workhorses of the cell. And so this particular process is really, really important for us to get to all of our proteins that are going to do all of the work in our cells. And so I am going to talk to you a bit about how post-transcriptional regulation also plays an important role in the regulation of gene expression as well. And the cell-specific expression patterns that we see in cells are in large part due to these post-transcriptional regulatory events as well. And as we think about post-transcriptional regulation, a lot of people in the field really think about capping, splicing, and providing the kind of the armor for this transcript to be competent to be used for protein for translation. But another side of this is decay. How fast you actually degrade these transcripts after they've lived a very useful life is very, very important to this regulation of gene expression as well. And I'm going to talk about a molecule, a multi-subunit molecule that's very, very important in processing and actual decay or turnover of these RNA molecules. And so there's a lot of information here, and I'm going to briefly just go through. The RNA exosome complex is in both compartments. As I mentioned, there's a nucleus in the cell and then also the cytoplasm. And the RNA exosome is a multi-subunit ribonuclease, which means it has the ability to actually chew up RNAs, process them, and actually degrade them. When talking to my students, I like to think about this. You can think about this as a trash can for RNAs. So it goes through and it can actually act to degrade RNAs, but also has features in processing RNAs as well. And so this machine is an essential, ubiquitously expressed machine. And so it means that it's in all of your cells in your body, and it is evolutionarily conserved and essential as well. And so it has a very, very unique structure. Importantly, it has a catalytically inert, which means it doesn't have any ability to kind of chew up the RNA transcripts, as I mentioned, from its core, its cap, and its core ring, but does have a catalytic disc 3 that actually associates with the barrel, the bottom of this barrel, which actually can act to degrade or turn over these transcripts. And so I'm going to get a bit more into why the structure is important in the next upcoming slides. And so what's very important, as recently as 2012, the first paper actually linked the RNA exosome to disease. And so mutations that encode these subunits of the RNA exosome have actually been implicated in a neurological disease called called pontocerebellar hypoplasia. And in pontocerebellar hypoplasia, you actually get wasting and atrophy of the cerebellum and the pons. As you can see here, the normal male has a very normal cerebellum, a very normal pons. And in this pontocerebellar hypoplasia patient, you have wasting or atrophy of the cerebellum and pons. And the pons is very, very important in your breathing, autonomic signaling, whereas your cerebellum is very, very important for muscle movement and coordination. And so in these patients, they actually were going home. The babies were born and went home normally, but would come back within weeks missing developmental milestones. So babies would present as floppy baby syndrome and a whole host of other developmental milestones they didn't meet. And so they did sequencing of these infants and then found the RNA exosome was the culprit. And another piece of this puzzle that's very interesting. And so remember, I said this particular complex is in every cell of your body. And so why is it that it's only affecting neurons per se, or that's the phenotype that manifests? And so that's kind of what my research gets at. Why is this RNA exosome complex, when there are mutations, actually affecting the brain in most parts? And another very interesting question here is that other subunits now that I've started to color in have been implicated in disease, and some of them are giving kind of different disease patterns. And so we're interested in why a complex with similarly situated and localized subunits are giving rise to different forms of disease that are a lot of times neurologic in its basis as well. And so 2 questions here: why neurons? Why is this complex in every cell of your body, but when mutated, it's affecting really very greatly your neurons? And so that's a lot of part of my research. A bit more about this pontocerebellar hypoplasia disease. It's of autosomal recessive inheritance, so which means it is both hits on both copies. So these patients have abnormalities or mutations, if you will, on both copies of the genes. And this is likely due to consanguineous marriages, first cousin marriages. So it's a very rare disease that was captured in the Middle East from first cousin marriages. And as I mentioned here, the wasting or atrophy of the cerebellum and pons. The pons, again, is involved in control of breathing and signaling. And then the cerebellum is involved in the control of muscle movement. And these patients actually also had developmental delays and intellectual disability. And so again, going back to why am I using the fruit fly model? Why is it relevant? And so again, we're thinking about why this RNA exosome, when you see a defect in one of its subunits, is giving rise to a neurological disease, and not all of the cells are manifesting with issues initially. Why is that? And so advance for me, please. And so we are interested in studying the fundamental biology. So we can use the Drosophila model to actually start exploring the tissue-specific functions or requirements of this subunit of the exosome to give us some insight into why is it that we see such a prominent neurological dysfunction or phenotype with these actual patients with these defects. And so there have been very productive studies in other model organisms too, such as budding yeast. We may have some budding yeast scientists or geneticists on the call. And we've all learned a lot from the budding yeast model, and now we're adapting that into the fly model because the flies have a really, really unique system to explore things in a tissue-specific manner. And so as I mentioned, why the fly model? So of course, the fly brain is not structurally analogous to the human brain. I think we all can see that, right? But what's really important to talk about here is that the processes which support neuronal function and signaling in neurons are actually very analogous in the fly. And so we can use the fly model to learn about these processes and gain insight into neuronal function and also morphology by using a lower model organism, a lower eukaryote, which can inform us eventually about the human brain as well. And so one particular structure that I'm going to talk about today in a couple slides of data is this structure called the mushroom body. So in the fly, we have this this structure here that's the learning and memory, the seat of learning and memory of the fly. And we have this prominent structure that bifurcates with alpha and beta lobes. And what we can actually do with very sophisticated molecular, genetic, and anatomical techniques, we can identify neurons of the mushroom body, which this provides a really nice biological venue to assess RNA exosome function. And so we can actually take away the RNA exosome subunit RP40 and see what happens to these neurons. And we actually can take the mutations that are in patients and CRISPR-Cas them into the flies and study what happens to these neurons as well. And so the first piece of data here that we were able to gain from our studies is that actually the subunit that's defective, that's causing this pontocerebellar hypoplasia, is actually required for proper mushroom body development in the Drosophila model. And so as you see here, this nice bifurcation and projection of the beta lobes medially and these alpha lobes dorsally, actually, you can see in our wild type flies, and these are confocal images that we use. We actually dissected the brains out of the flies and stained them with an antibody that's used for looking at enriched neuronal adhesion proteins. And so we were able to visualize these alpha and beta lobes of the Drosophila brain looking at these mushroom bodies. So this is wild type, or what we consider normal. We saw normal bifurcation and projection of these alpha and beta lobes. However, in our mutants, where we actually took away the RP40 subunit, so we actually depleted this subunit in neurons specifically to see if there would be any effect on the actual mushroom body structure, which would give us insight into neuronal morphology and function. And so as you can see, there were some issues. And so in some of our mutants, we actually saw this crossing over defect where the mushroom body, actually these neurons know where to stop. And so here you see the space here where there is no crossing or fusion defect in these beta lobes, because the neurons actually are programmed to know where to stop. And so in our mutants, we found very quickly that the neurons did not know where to stop and actually are fusing with each other. And also we saw some thinning of the alpha lobes. And so all of this is to say, this is our first clue that the RP40 subunit that's defective in these patients with pontocerebellar hypoplasia actually are causing morphological defects in the mushroom bodies, which can have a huge effect on function of these neurons as well. And so the latter part of my project that I actually wrapped up in my postdoc was actually using CRISPR-Cas9 to create Drosophila mutants. And so a number of missense mutations encoding single amino acid substitutions are found in conserved regions that are linked to PCH1B. And so I won't go through the schematic, but essentially there are single amino acid changes that are causing these changes and leading to disease. And a lot of people are familiar with sickle cell disease. This is similar to where you have a single amino acid substitution that gives rise to a disease. And so we use CRISPR-Cas9 editing, and people may be familiar with that. There's a lot of controversy about CRISPR-Cas editing and actually designer babies and things like that. But we can use this tool to actually take out the normal copy of this gene and put in the diseased copy into the genome, and then actually study its effects in neurons. And so we did just that. We CRISPR-Cas in these mutations. One of the mutations is a glycine to alanine amino acid substitution in this gene. And in these flies, we actually did see defects. And so you can advance one for me. And so in these slides, we see that wild type had the very nice bifurcation of the alpha and beta lobes. They projected nicely. They did not fuse at all. But however, in some of our mutants, we saw the fusion of these beta lobes, which led us to believe that just with this one single amino acid substitution on both copies, because remember, it's an autosomal recessive disease, we were able to see neuronal phenotypes where these beta lobes actually encroached upon each other or actually fused, which gave us another indication that RRP40, this subunit that's been found to be defective in people that have this neurological disease, there is a requirement that must be higher than other cell types. And so we're seeing this very neuronal like phenotype, but we're perhaps in other studies, we didn't see muscle phenotypes and we didn't see phenotypes in other tissues. It's particular or specific to neurons. And we were able to argue that these defects in RP40 are required for proper axon guidance and neurite extension of these actual mushroom body neurons. And so to wrap this up quickly, we were able to show that RP40 is required for proper development of Drosophila mushroom body neurons. And there's some other data I didn't get to share, but we also saw behavioral phenotypes as well as age-dependent lethality. So they had shortened lifespans and impaired viability. Then also I showed a RP40 mutant fly with a mushroom body defect. And so again, this study is really getting at why is this complex that is found in all of our cells giving rise to such a profound neuronal disease. And that is a lot of my study exploring why that is. And I think with that, we can wrap or take questions.
Yep, okay. Well, I understood the part when you said next slide, but everything else kind of was right over my head. So, but yeah, we do want to open it up for questions. I do want to mention again too, you might just, because the question that people always ask is, why is this work important and what is this work hoping to lead to? So kind of ultimately, what are you, in layman's terms, what are you hoping to find out through this research?
Great question. It's always good to think about that question. What is its relevance? Why is it important? And a lot of the studies that basic scientists are doing are really the foundation for how we even understand therapeutics. So we have the COVID-19 vaccine. Those studies started in the lab, right? And so it's the basic scientists that actually start the studies so those can inform later experiments, whether they're going to be in primates where you're studying things in primates, or are you going to create a vaccine? And so a lot of the basic scientists are understanding these very basic genetic interactions, or how cellular processes work, or how viruses invade a cell. What is it using? I think everybody's familiar with this spike protein that the COVID-19 virus, the coronavirus, is using to get into your cells. How do those dynamics work? And we use various experiments in the lab to understand that, so that informs therapeutics or other treatments or management or treatments of disease. And so this is kind of where we start, and hopefully my studies will inform therapeutics and eventually.
All right, let's go to some of the questions from some of the viewers. Let's see, Julie George has a question. Did you participate in research as a student at EKU? And since I'm the Dean of Libraries, did you use the library? Please say yes.
So I use the library all the time. I don't know if it's in this, if it's where it is, if it's in a similar spot, but I studied there all the time and used the library. And yes, I did participate in research with Marcia Pierce while I was there. I did a MRSA study with her in a student population on campus.
We have a question from Michael Earl Hay, and he says, it sounds like some amazing work that you have ahead with those grants. Are you involved with any such work to develop underrepresented population back here in Kentucky?
I am not specifically, but I am actually going to be staffing my lab and looking for trainees. And so I hope that I can come back to EKU and perhaps recruit some graduate students or some trainees to come over the summer to actually do research in the lab.
You mentioned the coronavirus, and obviously, if nothing else, this past year has taught us just how important scientists are. And I would think that as a scientist, it's kind of it's been somewhat gratifying and somewhat frustrating, probably at the same time. But I think this past year has shown us just how important scientists are, and scientific research particularly.
Yes, definitely. And also, I think for a lot of scientists, because there's a lot of misinformation or disinformation out there, I think it's very important that scientists come out of their labs and share their work and actually talk with the community about what's going on. And I think there's just a disconnect there as well, and really a lot of misinformation about what we're doing in labs and how we're coming up with things such as the vaccine, et cetera. And so, excellent point.
As you look ahead, are there goals, things that you still want to do? What are some things that you are thinking about in the future for Dr. Derrick Morton?
So my immediate goal is getting my lab up and going. I'm very, very passionate about training programs. And so I'm launching one this summer. Very small pilot program that was funded by Burroughs Wellcome. So my goals are training in the lab students, making some really cool scientific discoveries, and hopefully gaining promotion and things like that at my university.
You think you're going to stay in California for a while?
I'll be here for a while, yes.
Do you get to make it back to Kentucky much?
You know, I visit my family. My mom would want me to visit a bit more. But I try to come back definitely for Christmas. And I try to come back at least once some other time in the year.
Well, we certainly hope we could get you back on campus sometime and show you it's changed a lot since your days here, and you may not recognize it. But we would love to have you back on campus, and we certainly have enjoyed having you tonight and appreciate you spending some time with us.
Yes, same here. Thank you for inviting me.
Dr. Derrick Morton has been our guest tonight on the Alumni Spotlight Series. Dr. Morton, thank you very much. And we appreciate not only your research, but it's amazing to me. I'm proud to say as a fellow alum that this guy went to EKU, and look at how smart he is.
Thank you. You all have a nice evening.
Dr. Derrick Morton, our guest tonight on the Alumni Spotlight Series, again, as part of Scholars Week this week. And we thank those of you who are tuned in tonight as part of Scholars Week. Again, a lot of other programming coming up that we want to let you know about in the next couple of weeks. Later this week, we have the At the Races event, the virtual racing night. We want you guys to join us Thursday night at 8 o'clock. All alums are invited. Anyone who would like to come and join us is invited. It's virtual day at the races, and we would love to have you join us for that. Also coming up on the 14th, and Dr. Wade mentioned it a minute ago in the comments, EKU's Giving Day. 1 day, one goal, 1,000 donors, and we have future Derrick Mortons who are on this campus right now and could use your support, and we hope that you will consider giving and supporting EKU on Giving Day on the 14th. On the 15th, it's our next bingo night. The Student Alumni Ambassadors present Alumni Bingo, 8 o'clock on April 15th. We hope that you will join us for a night of fun and fellowship for that. And then a couple other programs. Our next Spotlight series is coming up on the 20th of this month. Dr. Deborah Alexander, with a distinguished career at the State Department, she will be our guest and talk about her many life experiences. She did a great photo exhibit on campus. It seems like last year, but with the pandemic, everything seems like last year now. It's probably been a couple of years ago now, but fascinating career. And I look forward to talking to her 2 weeks from tonight at 7 o'clock on our Alumni Spotlight series. And then we will have the Colonel's Kitchen, a special Kentucky Derby edition of the Colonel's Kitchen. That'll come up on April 28th at 8 p.m. And we hope you'll tune in. Again, that is a joint production with the Department of Applied Human Sciences. Dr. Dana Bush and myself will be your hosts for the Colonel's Kitchen on April 28th. That will wrap up things for us tonight here on the Alumni Spotlight series. We look forward to seeing you next time. Until then, from the Campus Beautiful, I'm Dan McBride. Good night.